So I have Dr. Viraj Main with me tonight, and he is co-founder and chief scientific officer of a company called Lactiga. So Viraj, go ahead and pull up your slides. I have to say, as a pediatrician, that this in and of itself is fascinating to me. How you came to develop this new therapeutic. So can't wait to hear more.
Thank you, Katie. It's such a pleasure to be in a room with you again. So by way of introduction, Lactiga is a company that's actually incorporated in Toronto in Canada, but my partner and I currently live in New Jersey. So we do have a footprint in 2 different countries. And today I'm really excited to share with you how we have built this company to protect patients with primary immunodeficiency diseases against mucosal infections. So the category primary immunodeficiency diseases actually constitutes over 400 diseases, many of them quite rare. These are life-threatening conditions with a dearth of solutions. Now, all of these patients in this category are vulnerable to infection pretty much at all times. And I'm not talking about sniffles or cold or mild infections, but rather severe and recurring infections that put these patients in and out of hospitals. Now, for the specific PID disorders we would initially target, that already accounts for over 100,000 Americans with specific PIDs that would need treatments. They experience exacerbated morbidity and mortality, especially manifested as recurring mucosal infections in the respiratory and gastrointestinal tracts. There's a very short list of existing treatments, which are expensive, invasive, painful, and have a long list of side effects, which can include nausea, vomiting, diarrhea, severe pain at the site of injection, and extreme lethargy. Most of these medications are delivered by infusion or injection, as you can see in some of these graphics. And those infusions can take up to 8 hours, and that's going to happen at least once a month for the rest of their lives. Now, as we think about additional conditions where severe mucosal infections are prevalent, that includes several cancers, such as lung and gastric cancers, as well as AIDS. And that stacks an additional 1.8 million Americans on top of our first go-to-market indications. So to address this dearth of existing treatments, we are developing our patented solution, which contains dimeric IgA antibodies, and these are derived from human milk or breast milk. And that's because human milk is naturally enriched for dimeric IgA antibodies, which are the only antibody subtypes known to be stable in the mucosal compartments. And you can see their dimeric structure in that small cartoon. These are highly polyclonal, which means they provide coverage against all forms of pathogens, viral, bacterial, fungal, parasitic, etc. Now, human milk is the only scalable source of dimeric IgA, and for that reason, we have already built an international supply chain, the world's first, involving the global network of nonprofit milk banks, which I'm happy to share more in the Q&A. In addition to leveraging these interesting properties of IgA antibodies, we're also developing novel delivery solutions you can see in the bottom half. Specifically, we're developing these into respiratory formulations, and you can see an example of a home nebulizer in this photo, as well as oral products for gastrointestinal delivery. That makes our products differentiated, low-cost, easy-to-use adjunctive therapies to existing treatments that would not require needles, phlebotomists, or any type of specialty care. Put simply on the bottom, IgA milk antibodies can do what serum antibodies cannot. And this is supported by pretty extensive in vitro validation we have completed. So across the top half of this graphic, we looked at the ability of milk antibodies to neutralize RSV, or respiratory syncytial virus, a major driver of a severe common cold. When we first looked at cellular neutralization using milk antibodies, we determined 85% neutralization of RSV. When those antibodies were frozen and subsequently thawed after 3 years to retest them, they retained 64% neutralization. Additionally, we looked at room temperature shelf stability, which is on the top right, and for at least 2 weeks, they can sit at room temperature with no reduction in stability, really telling you about the durability of these milk antibodies. Now, across the bottom half of this chart, these actually reflect our SARS-CoV-2 datasets for COVID-19 in conjunction with our partner at Mount Sinai Hospital in New York City, which is supported by a large NIH grant. And these figures on the bottom are actually from a peer-reviewed publication. And specifically, we looked at the presence of COVID-specific antibodies in milk samples from COVID-infected mothers. 100% of those milk antibody samples are capable of binding the novel coronavirus spike protein. In the middle, you can see that 80% of those samples also bind the receptor binding domain, or RBD. And even more importantly, on the right, those antibodies are capable of 100% inhibition of cellular entry. So, very compelling in vitro data set. But what gives us the confidence to believe that this will translate to a successful nebulizer delivery program? Well, there's two layers here. First, in terms of the existing background, as I mentioned, milk is known to have a natural abundance of dimeric IgA antibodies. If you look at the red circle on the top right, you can appreciate how different these are structurally compared to other antibodies. And that makes them uniquely stable and bioactive in the respiratory tract. But in addition to that, airway delivery of these IgA antibodies is in fact a de-risked concept with significant stakeholder support. And what I mean specifically by that is that we have a research collaboration with Mount Sinai Hospital, as well as an exclusive commercialization agreement with them. This is supported by a $2 million NIH grant, plus 2 other cash awards, all in support of our nebulized COVID-19 treatment. We took our technology notes and submitted a pre-IND to the FDA last summer and secured their written feedback to further guide our COVID-19 nebulizer program. And thinking more broadly, there is in fact several pieces of peer-reviewed literature about the anti-infective properties of IgA in the lungs. For example, this example of neutralizing Pseudomonas aeruginosa, a severe respiratory infection in the mouse lungs. And lastly, on the bottom, antibodies have also been nebulized and traced in the primate lungs. Taken together, IgA antibodies are able to be delivered and are highly effective in the mammalian respiratory tract. So on this slide, I'd like to give you a brief overview of the company history, drawing your attention just to the blue headings across the top. So as an early self-funded company back in 2017, we really focused on patent-enabling research engagements with top-tier research institutions. Last year was a big year to open up our COVID-19 program, execute the collaboration with Mount Sinai, and open milk supply agreements in 3 different countries, the US, Canada, and the UK, while also having our Canadian patent issued. Now this year, 2021, has been quite massive for us because we've moved from early proof of concept into the actual manufacturing engagements represented by the logos you can see here. Not to mention we had our US and Japanese patents also issued. So by focusing on the manufacturing, which is our current priority, that allows us to transition into non-clinical efficacy, leading to an IND submission in the year 2022, which ultimately allows us to conclude a phase 1 clinical trial in the year 2023, which is the final deliverable you can see on this slide. So who is the team to execute on this highly transformative and ambitious vision? Well, speaking for myself, I have a PhD in human genetics and 5 issued patents, so I'm leading the IP and research portfolios. Now, the perfect complement to my skill set is found in my co-founder, Rick and Mehta. He's a former FDA official. He has held senior roles at Pfizer and Aquestive, so he not only has regulatory expertise, but also a corporate pharma background. For the larger Lactiga team, you can see on the bottom, we have quite a deep stack of credentials. We have a medical advisor who actually treats the primary immunodeficiency patients I've been talking about. We also have a roster of research collaborators with highly specialized skill sets relevant to our work, which has helped us access NIH funds as well as other programs and has led to 2 institutional commercialization agreements. Finally, we have KOLs who are the chief executives of North America's largest immunodeficiency patient foundations, whose logos you can see at the bottom. So with that, I'll stop here and I really look forward to your questions and feedback. Thanks so much.
Viraj, thank you again for being here and sharing with us about Lactiga. This is really exciting. You know, one of the things, that I think about in my mind, like there are a lot of moms out there having babies for sure. And it sounds like you have some good partnerships with these milk banks, which is very cool. But how do you know this is scalable, right? I mean, is it sustainable through development, production, et cetera?
Indeed, it's a great question. And so people typically assume like, well, there can't be that much out there. And even if there was, why would they give it to a company like yours? And so here's some of the stats that support our scalability. So there are over 700 milk banks around the world. They primarily function as a large network of nonprofit institutions. About 32 of them are in North America. We've already opened supply commitments, as I mentioned previously, in 3 countries. And we were able to do that because we positioned a story about bringing novel medical benefit to highly vulnerable patients in a way that aligns with their mandate. Now, their mandate is, of course, to focus on medically fragile babies, especially premature babies. By being very sensitive to make sure we could support their core mandate, and never interfere with eligible milk that they needed for their purposes, we could instead focus on the unused fraction of their volume. And by the way, each of these institutions can discard up to 15% of their supply because it doesn't meet their inclusion criteria, but it does meet our inclusion criteria. And so just to give you a snapshot of what just today's small handful of commitments already gives us, it puts us in the ballpark of dosing 10,000 to 30,000 patients per year, just using today's commitments, which represent 4 or 5 entities. Again, that means there's about 695 other milk banks we could still tap. And the early champions of milk bank directors have been extremely vocal and proactive about reaching out to their colleagues on our behalf, sometimes without us even being in the room. They're so motivated, they're actually having these discussions. So we are fully confident that the supply network will only grow simply because we have focused on alignment with their core mandate before we ever started opening this discussion.
Sure. No, that sounds awesome. It sounds like you have done your homework and definitely approached this in the right way, which is great.
We had to be very sensitive, knowing the audience.
Yeah, for sure. So one other question that I have, just from a perspective of, you and I have talked about a patient actually that I was involved with, peripherally involved with, I would say, because this was an adult patient recently with a severe immunodeficiency. The patient, story that you gave actually during your presentation. And what I know is there aren't a huge number, right, of those patients that are out there. So it seems like you're starting with a smaller market in the grand scheme of things, but I am guessing this same technology can be applied to other use cases and other larger markets. So talk a little bit more about that.
It's a great question. There is an assumption more broadly that if you're targeting a rare disease, there are so few patients, you'll never make much revenue as a biotech company. That's doesn't align with the various different rare disease incentives that are out there. And I have another slide, maybe we can touch on it in a second. But to give you a sense, Katie, you're absolutely right. There is the primary immunodeficiency cohort on which we really developed this company because of how closely these solutions would match their needs. And that's the addressable market we're showing on the left. So, and this is just in the United States. So we're looking at around 100,000 PID patients, which are highly symptomatic because of selective IgA deficiency or common variable immune deficiency. With our target price, revenue price of $25,000, treatment revenue per year per patient times 100,000 patients, that's a $2.5 billion figure. As I mentioned very briefly in the presentation, as we expand outwards, it will be logical to address other patients who also have frequent recurring and severe mucosal infections. Right off the bat, that includes patients with gastric cancer, lung cancer, which is this middle bucket, and then AIDS patients, of course, are also afflicted by severe and recurring mucosal infections. So this figure, or this slide by itself, is already a $10 billion figure. And these are just 3 of the longer list of indications that we would ultimately target. And to your point, this is absolutely a platform technology. As we develop out the respiratory formulation and then the oral formulation, those can be, we can apply for additional indications without really having to reformulate them or to redesign them. The goal is to use the platform technologies as broadly as we can.
Well, that's fantastic. Viraj, again, thank you for being here tonight. I know there are a few questions in our Q&A from the audience that we did not get to, but during the networking session, I'm sure Viraj would be happy to take those questions. And I just, again, exciting company. I love what you're doing. Thank you again for being here tonight.